Hello and welcome back again. This is Ms. Bushra with you, and we're back again for another episode. Today, we're going to pick up where we lived in the respiratory system, section 3 of chapter 2. We talked about the lungs and how many lobes are they, and now we're going to check how the lungs actually going to exchange gases. Remember, all we talked about it is that the place where all exchanges happen in is actually alveoli. This is how alveoli is empty. The air is coming inside to these smaller bags, and then it's surrounded by these networks of capillary. If we magnify a very small space where the alveoli wall and the capillary wall meeting each other, this is the part of alveoli, and this is part of capillary. You will see that they are so thin that it's so easy for them to exchange materials between them. Remember, we want oxygen inside the body, so it goes to the inside of the capillary. Why is that? Because the content of oxygen is high in the atmospheric air that reaches the alveoli, while it's very low in the blood that is deoxygenated, obviously, because the tissue cells have used it. Now, same information. The tissue cells that use the oxygen, they release the CO2 within the blood. Now, the content of the blood CO2 is much, much, much higher than the atmospheric one. So, naturally, by diffusion, the CO2 goes out of the cell. Let's see. In the gas exchange, two more information that may bring you back. CO2 needs to be get rid of because it's toxic, and oxygen is needed for respiration for making ATP. Just to remind you. So there you go. They're in opposite direction like we said. The alveoli is high in oxygen, low in carbon dioxide, while the blood capillary is low in oxygen, high in carbon dioxide. We're going to talk about how these two are actually transporting. Remember we said like it's diffusion because of diffusion and their direction, we already said it. Let's see how oxygen is transported. Now oxygen is transported in two ways. Mainly, we talked about this so many times that you should remember it. It's going to be transported by red blood cells because red blood cells have a certain type of protein called hemoglobin. There you go. Hemoglobin, also known as HB. Hemoglobin is going to grab about 95% to 98% of our oxygen, and it carries it into the destination. So according to our information, it should be like that. However, the smaller amounts remaining are just going to dissolve in the plasma. Very small amount, like about the opposite of that, 5 to 2%. Only this much. Remember, hemoglobin is going to grab onto the oxygen, since the hemoglobin, if I remind you again, it is 4 parts, and it's a globular protein which is made of 4 parts. Each part contains an iron. That iron is able to hold on to oxygen. Each one of them an oxygen molecule, like that. So each iron is holding one oxygen molecule. Because we have 4, each hemoglobin is holding on to 4 molecules of oxygen. Where does that happen? This happens, okay, this combination happens where it becomes HbO8, that's called oxyhemoglobin. It happens within the capillaries surrounding the alveoli. Now it's ready to be transported. This is like how it's packaged. We're going to take it to the tissues. Every time it goes to a tissue, we need to let go of the oxygen so that the tissue can take it. So since we have like this reversible mark you studied in chemistry, it is not, the reaction is going to be reversed back. There you go. We have HB, 4O2, 4O2, and this is going for the tissues. Where does this happen? HB breaks down, HbO8 breaks down within the tissue, while HbO8 made in the capillary surrounding the alveoli. That's how you're going to describe it. There you go. There's like a quick review if you want to. Everything I just mentioned in a very short method. Okay. What about carbon dioxide? Carbon dioxide is totally in the opposite direction, and the way it is transported is actually not 2 ways but 3 ways. Let's see. The first way, which is the least common, is just dissolving in plasma. These gases, when they dissolve in plasma, they're not that much, both of them. They're the least amount. And then approximately about like 23% is going to attach to hemoglobin. It's going to attach to hemoglobin. It's not natural for it to do that, but it's able to do it. So 23% of it. The remaining 70%, because the dissolved one was 7, the remaining 70% is going to be transported in a shape called bicarbonate ions, HCO3 minus. Bicarbonate ions, you know too well that carbonic acid, this is carbonic acid, okay, it's a very unstable and weak acid made from H2O and CO2 reaction. It's going to get split, and then it gives us what? The bicarbonate ions and of course an H plus. So in this case, where does this happen? It happens within the tissue. In the tissue, when we want to get rid of the CO2, it goes to the blood capillary surrounding the tissue, and it gives us the bicarbonate ion. This is a very good form to send it back to the lung. So once it's in the lung, the bicarbonate reaction, again, it's reversible. Let's change the color. This is the bicarbonate ion. It's going to react with the hydrogen again, giving us the carbonic acid. And then it's going to get reversed back to H2O and CO2. This is how it goes out of the lungs. That's it. That's all you need to know. You need to know where does the reaction happen and where does this one happen. When does the HCO3 was created and when is it going to get broken down and react back to its original phase? These are very important to know where it happens, and this is a quick review regarding this. The mechanism of breathing. It's super easy. We need a bit of physics here, but it's going to be easy if you focus on what I say. The first thing I want you to know is that when you breathe in and out, this is called breathing like we know. However, when you do this, it's inhalation, there you go, or inspiration. When you do this, that's expiration or exhalation. All of them are fine, as long as you know that the air is going out. However, don't mix it up with external respiration. That's a totally different matter. It happens with the blood, not just going in and out of lungs. That's not what it is. So remind yourself with the other one so that you don't get mixed up between these things. Let's see how this happens. I want you to know something, a physics law. Let's change the color. It says when the volume increases, volume of a space increases, the gas pressure decreases, and vice versa. What's that supposed to mean, Miss Bushra? It means if the volume decreases, the gas pressure increases. Remember that they're inversible, okay? They're not directly proportional, but actually inversely proportional. So if you check this, you know that our mechanism of breathing is not with a pump. Nothing is going to bring air by force, but actually is a trick. How's a trick? It's because when you do this, when the brain sends the signal to the muscles, remember, brain can control the muscles and the glands. So when it sends it to the muscles and the muscles are starting contracting, when you do this, there will be more space in the chest area. More space means lower pressure. And air like something. All gases going from high pressure to low pressure. If now my lung have lower pressure than outside, so naturally the air comes inside. That's not because you did it on purpose, like forcefully, but actually because the area here got expanded so much that the air comes naturally. And then what about when you exhale? The muscles are going to relax back, bringing the chest down. And of course, we're going to talk about diaphragm later, but remember, the area in your chest cavity is going to get smaller, the lungs getting smaller, and the air is going to be forced out. It's all physics. It's all physics. Let's talk about what muscles are actually contributing in this. Between the ribs, there is a type of muscle, we named it after them, the rib muscles. And below the lungs, which separates the chest cavity from abdominal cavity, we have diaphragm. This is diaphragm. Tamam? Now, when you inhale, the chest cavity gets contracted and shorter, lifting up the rib cage with it. So the rib cage now is lifted. Lifted not only upward, but a bit outward too, creating space vertically and horizontally. And of course, the diaphragm was pushed down because diaphragm naturally, on relaxed state, it's like this. It's a dome shaped. Okay, it's curved up. This is like the nature of the diaphragm. It's curved up. This is flattened. It goes down and get flattens. It creates more vertical space. So this creates more horizontal and vertical space, while this one is creating even more vertical space from the downside. This creates like a bigger space within the chest cavity, making the lungs, which are elastic, to expand. Once they expand, the law applies. The air naturally follows to inside, and that's it. Opposite of that, when the rib muscles and the diaphragm relax. Remember, diaphragm is flatter during contraction. It is dome shape or curved shape when it is relaxed. While this one, rib muscle, when it get relaxed, it leaves, okay, or puts the rib cage downward like that. It's shrinking the entire space, making the lungs shrink again because it's elastic, like I said, like a balloon. And then the air naturally just goes out. But Miss, how do you remember all this? Of course you will remember it because I put it into a table. So there are some details here. The diaphragm, which is a large skeletal muscle, first separates the thoracic from the abdominal cavity. Secondly, it's also part of what? The respiratory muscles. Respiratory muscles are two, the rib muscle and the diaphragm. Contracting them is going to make the person breathe in, and when relaxing, it's going to breathe out. Of course, the lungs are elastic, like we talked about it. It can expand just like a balloon and then recoil like a balloon too. The differences of pressure, it's what brings the air in and out. Let's not forget about that. Just how when nutrients are moving in and out the blood, it's because of their concentration gradient. This is because of their pressure gradient. There it is, the table. The table holds everything here. We can go over it one more time. In the inspiration, both the rib muscles and the diaphragm contracts. When they contract, what do? They increase the space within the cavity, in the rib cage, and of course the lung is going to increase with them. They are not the same thing, by the way, okay? Now, the thoracic cavity is different. It's the space, but the lung is the tissue that's getting expanded with the increased space that available. Remember, increased volume means decreased pressure. This is how the air is pulled into the lung, because compared to outside, the inside will have less pressure and the air naturally goes from high to low. It's opposite in the breathing out. The space is becoming shrinked, and that shrinking means less volume. Less volume in the lung means higher pressure. So naturally, air is higher in the lung, goes out. Because outside doesn't change, but the lung change the pressure in the lung. When we regulate the breathing, remember it's all about the activity. The faster the activity, the faster the breathing. The slower the activity, the slower the breathing. Why? Because it's about demand and take. If you are doing more activity, there will be more demand of oxygen to work out and gain more ATP, just like when you exercise. But during sleeping, the activities of your body and muscles are going to be reduced so much that you don't need this much of oxygen. That's why naturally your breathing adjusts to the need. The body is smart. It's never going to consume something more than needed or less than it's needed. It's always like regulating just the right amount. Now, the rate and depth of your breathing is regulated, like we said, to provide oxygen at the same time to eliminate carbon dioxide. Because too much carbon dioxide is toxic, and it shows that actually there was a lot of ATP used and oxygen consumed in the process. This is going to be the start of this question. So, how does the body, or which part of the body regulates the brain and mainly your brainstem? Brainstem monitors means keep it in check. When you say monitors, it means it has a sensories that can detect carbon dioxide presence in the blood. Now, as the activity increases, high level of carbon dioxide are going to stimulate, means signal, the nerve cells in the brainstem, which in return stimulate the diaphragm and the rib cage to start contracting and relaxing much, much faster. This carbon dioxide concentration in the blood, if it gets lower this time, this is how the breathing gets lower. So at the beginning of an exercise, your breathing is natural, but then your muscles use more of the oxygen, which makes more of the CO2, and the concentration of CO2 is what is going to tell your brain it's time to breathe faster. Okay? It's not about oxygen. The brain recognizes CO2. It says, can an individual consciously stop their own breathing? Yes, temporarily. Only temporarily can override it. How? Because it's actually like you say, let's say it's on autopilot mode usually, but you can take it, especially during swimming. For instance, you can hold your breath for a while, but then if you do it too much, your brain knows about it and it will knock you off and you're becoming like unconscious to gain that oxygen back. So it's just for a short period of times. Diaphragm is so important that paralyzing it, it's going to stop the entire breathing process and it kills the person. So it's very, very important for the diaphragm, okay, to keep moving on so that and doesn't get paralyzed. One of the toxins in the foods, like called botulinum toxin, that we're going to study it in chapter 3, it is actually working on, paralyzing muscles. The moment it reaches the diaphragm, it's done for. And this is how we're going to start our chapter 3. Chapter 3 is very, very nice. It is all about the body immune system. We're going to talk about the non-specific immunity in section 1, specific immunity in section 2, and of course, we're going to talk about something so challenging for the immunity system, which is the HIV. Let's start. We're starting section 1, which is non-specific immunity. The non-specific immunity, we need to know two things before we start the whole chapter. The first one is what is infectious diseases, and the second one, what is a pathogen. So infectious diseases is any diseases that caused by a pathogen. But what is a pathogen? It's an agent that causes the disease. Usually it's a microorganism. Could be a bacteria, a fungus, a virus, a parasite, a protist, whatever it is. infectious diseases usually are the one that get transmitted and could be, exchanged between people just because they touched each other, or like, for example, from their own food. An example is rhinovirus, which is a type of virus that causes common cold. Look how fast it is transmitted between people when you get cold, when they get like, flu. These things are all infectious diseases. About infectious diseases, there was like a very smart doctor called Robert Koch. Robert Koch, this is his picture, actually wanted to know what causes these diseases that could be transferred between people, and sometimes not only between people, between animals and people. So he did something. He created a step-by-step procedure called Koch's postulate. This is what we call now. This step-by-step procedure it was used for identifying pathogen. Aim or objective, it is identifying pathogen. Especially he was studying on something at that time, anthrax. It's a type of infectious diseases. He showed that we get it from cattle. Cattle means cows, sheep, those types that they kept in the farm. It's spread to people. So he saw that these cattle illness had swarms of a bacteria that had a rod shape. So there was a bacteria that has a rod shape, this is rod shape, in their blood. So if that's the case, he wanted to know whether this is actually the cause of disease. So he developed a four-step, procedure based on his hypothesis. Hypothesis because he thought it was not actually like today. Like the science were not that advanced to know how to deal with these stuff. He designed all this. Until this day, we're going on this method because it's so useful. The first thing he did, he isolated the rod-shaped bacteria that were from the blood, okay, in a cow with anthrax, so a sick cow, okay? A sick cow. What did we isolate? A bacteria. Isolate means like we sampled it. Tamam? Then he took that sample from the blood and he put it on a petri dish and grew colonies. Remember, petri dish is these dishes in the lab where you strike it like this in order to get a growth. If there is bacteria, they will grow on it and you will clearly see it. And what he did, he saw the growth and took the ones that he thought this is the one that is causing it and isolated a single species. Because the first sample you get, it's a mix of a lot of species. So isolating it based on each one species, he prepared injections. What did he do? This time he brought healthy cow. Remember, in the first one we had a sick cow. In the second one we grew things in the lab. In the third one we brought a healthy cow and started injecting it. You need to know all these keywords. Injection is in third, Koch's postulate, while working in lab is in second and in fourth. There you go. He injected it to a healthy cow. He saw that these bacteria actually developed anthrax. It was the bacteria who caused anthrax. To further analyze and make sure exactly this happened, what did he do? He checked that the same bacteria was in the first cow was exactly here and same disease was developed, which is anthrax. And in the fourth one, the healthy cow that were not injected at all, they did not develop anthrax. They used this as a control group. Okay, this is how his method was proven to be effective and named it Koch's postulate as a hypothesis to see which agent is actually causing this disease. These four rules, until now, they're using it. For corona, they use it for everything that is not found yet, okay? For identifying the cause. There you go. These pictures might come back, actually highly possible, what did he do first? He first isolated a pathogen from animal. He saw that a diseased one has it and a healthy one did not have the same bacteria. That's how he knew it was the cause. Okay, and then to further confirm it, he isolated and grew it in the laboratory, and then he injected the what we had into here. It says it's mice, but in his original work it was cow. However, it means the healthy animal. In here, he again worked in the laboratory and saw that the second animal has actually the same pathogen like the first animal. First animal from first step. Second animal, we mean the one we injected in the third step. That's the second one. What are pathogens? Pathogens are microorganisms. Could be a bacteria, could be a virus, could be protist, could be fungus, could be an invertebrate. All these things that we already studied. How do they get transferred? Naturally, by air, by water, by food, anything that could be shared, by towel, by clothes, anything that could be by touch, like we said, or direct touch, even. Okay, even bites of animals. If you notice, all this was, a method of transferring in corona time. So I'm sure you know about it because we've been through a pandemic. This table, I want you to know it just like your name. There it is, examples of each one of the type of pathogens that I mentioned it and what they are causing. Like, for example, a very well-known sickness. A botulism is a disease caused by a bacteria called Clostridium botulinum that you get it from contaminated food, especially canned food. Like, for example, canned children food, like milk, whatever. If you check it and it gets inflated, that means there was gas produced because there was a growth of bacteria. You should not eat it. AIDS, like everybody's nightmare, comes from a virus called HIV. So the AIDS is the disease. HIV is the virus that causes AIDS. Okay, from sexual contamination, from any fluid, body fluids that get shared between them, like through needles, through other fluids, mother and fetus, everything. And another one, amoebic dysentery, which is like a diarrhea with blood situation, comes from Entamoeba histolytica. We studied this in grade 11. We don't need to elaborate on it. Food and water were the source. Athlete's food, this is where somebody is like exercising or they wear their shoes and socks and there's a lot of sweating or they were like running in water. There is a dampness and they are in their shoes for a very long time. They might get a type of fungus called tinea that could be transferred between people if they shared shoes, towel, or socks, or anything that could be like transferring it. But mainly those person to person contact or like a specific thing that was not supposed to be shared, they shared it. Non-specific defenses. Let me tell you, we have in our immune system or like our body protecting us in two ways, either specifically or non-specifically. What's that supposed to mean? When it's non-specific, even if they don't recognize which type of pathogen is this, was it the botulinum toxic one, was it Clostridium, was it tinea? No, we don't care. As long as it is something that our body knows it's not from us, it is from outside, it is a foreign body, it's going to attack it, regardless of the identity of the pathogen, like here it says. Non-specific defenses is for next lesson, but let me give you a heads up. Skin and mucous membrane are the first barriers. HCl kills bacteria. Inflammatory responses are also non-specific. Fever, which is high temperature, and some proteins in our blood system. So next time, inshallah, we're going to start here, and I will see you then. Have a nice day.